And at the end of each question period, we will have the opportunity for you to ask questions. So feel free to use the chat function or the raise hand function to do so, and we will unmute you. And with that, I'm going to pass it on to our first speaker, Casey Williamson from the University of Guelph. And today she's going to be presenting her work entitled Loss of Nephrin Phosphorylation Impairs Podocyte Force Transmission and Exacerbates Diabetic Neuropathy. And with that, I'll pass it on to Casey.
All right, thank you for the introduction, and thank you for everyone for joining today. I'm just going to get set up here. All right. Okay, so my name is Casey Williamson, and today what I'll be talking about is how nephrin phosphorylation can impact podocyte force transmission and how this relates to diabetic nephropathy. I'm a PhD candidate at the University of Guelph in Nina Jones' lab. There we go. All right, so podocytes are some of the most susceptible cells for kidney injury. When looking at a kidney here, the interface between the medulla and the outer edge of the cortex, there are roughly 1 million nephrons in the kidney. And these nephrons, when looking at the actual vasculature, they look like little tufts of capillaries. And this tuft of capillaries, when actually looking further, there's these specialized cells known as podocytes that wrap around the capillaries with these interdigitating processes known as foot processes. When taking a cross-section or a section of this, shown here, the foot process is right over here, and then in between them, there is a molecular filter known as the split diaphragm, and the podocytes then sit on the glomerular basement membrane. And this is what it looks like when they're healthy podocytes. However, when they're injured, they essentially create this change in structure that's known as foot process effacement, shown here, with a loss of split diaphragm density. And also the glomerular basement membrane can also be thickened. Now, these changes are also associated with many other kidney changes, including glomerular sclerosis and hypertrophy, alterations in glomerular filtration rate, or GFR, and then this can then progress to the detachment of podocytes into the urine, also known as podocyturia, and this is irreversible for the injury since they can't divide, and this also could then result in the loss of protein or proteinuria. So there's lots of factors that can affect podocyte injury going further. Some of these factors I'll just kind of highlight right over here: growth factors, hormone dysregulation, cytokines, high glucose, such as in diabetes, and lipotoxicity. However, one of the most underappreciated factors for podocyte damage is mechanical strain. So shown, as mentioned, glomerular filtration rate can change, and this can lead to glomerular hyperfiltration, which can then progress to that podocyte detachment. To highlight some of these mechanical changes, here we have a capillary, and the filtrate goes right through the slit diaphragm, and foot processes are here. They also then resist shear forces, such as shear forces at their basolateral domains and shear forces that are apically applied as well. And there's also, through the actual capillary pressures, hemodynamic pressures, there can also be tension forces as well. What's really important to note is in certain pathologies, such as diabetic nephropathy, the actual change in mechanical strain can precede proteinuria. So GFR is shown right over here, and in diabetic nephropathy, for example, GFR goes way up, which is essentially hyperfiltration and higher mechanical strain, and the proteinuria then is successfully after that. And as you lose more of the glomeruli, some of the remaining glomeruli have to then they have more mechanical strain to actually compensate for that kind of filtration. So there's actually more mechanical strain as the disease progresses. So this is something that's really important to note for podocyte pathology, kidney pathology, and going further, podocytes have several adaptations against this mechanical stress. So for example, there's strong cell-cell and cell matrix adhesions in podocytes at the slit diaphragm and focal adhesions. To show this here, we have slit diaphragm membrane proteins between them, the focal adhesions shown here, and they then create this dynamic actin regulation as well. The central slit diaphragm protein known as nephrin might have roles in mechanical signaling, and this can then be carried out through signals, for example, through phospholipase gamma, which can then signal to calcium signaling, and calcium can then control some of the actomyosin signaling as well, or contractility. Through NCK adapters as well, there can be dynamic actin regulation, hippo inactivation through YAP, and then there's also could be potential crosstalk with focal adhesions as well. However, nephrin's role in mechanosignaling or podocyte force transduction remains uninvestigated. To go further into nephrin structure in the slit diaphragm, there are these specific tyrosines within YDX3 motifs that are very important to note. So at the slit diaphragm, nephrin can actually undergo a clustered conformation, which can induce this phosphorylation. So to show this right over here, there is a lone nephrin molecule, and this is the top view of the slit diaphragm. And under certain circumstances, nephrin can undergo this transinteraction that creates kind of like a zipper pattern, and this zipper pattern can then lead to the phosphorylation of these YDXV motifs. And through these YDXV motifs, they can signal to some of those aforementioned potential molecules involved in mechanosignaling, such as PLC gamma and NCK adapters. And in some of the previous studies on this, we find that there's increase and decrease YDXV phosphorylation in several podocyte injuries. However, some diseases might have an increase, some diseases might have a decrease, and elucidating the actual signaling mechanisms can be a little bit tricky. However, we then hypothesize that nephrin YDXV tyrosines signal downstream mechanical changes in podocytes, and that these residues are crucial for mitigating mechanical strain in podocyte pathologies. So for our first aim, we used a CD16 clustering system of nephrin to assess podocyte force transmission in vitro. We used a immortalized mouse podocyte cell line, or MPCs, that differentiate approximately over 14 days, and then they exit the cell cycle and create that specialized. They don't necessarily create slit diaphragms, but they do have more of their differentiated features. We then transduce them with a fusion protein, which is known as CD16-7 nephrin GFP. And this fusion protein shown here, it has two extracellular domains of CD16, a CD7 transmembrane domain, and then the cytoplasmic region of nephrin tagged to GFP. And when you apply a CD16 antibody, it can then induce this clustered conformation that induces nephrin phosphorylation. We then also transduce these cells with a CD16-7 nephrin Y3F GFP fusion protein as well, which does not have that same capacity for phosphorylation. We then assessed for several signals involved in contractility, so actomyosin contractility specifically, and one of the primary methods of contractility in podocytes is non-muscle myosin 2A, and we assessed myosin light chain 2 for this type of metric. And then we also assessed the filamentous over globular actin ratio as well to determine how much of that actin polymerization is actually going on and how stable it actually is. We then also applied this methodology known as elastic resonator interference stress microscopy, or ERISM. And to explain this methodology, MPCs were seated on a collagen 4 coated gold sheet. And then there's an elastomer that sits on, and it's sandwiched in between two gold sheets, another gold sheet here. And whenever podocytes engage in a pulling or pushing force, it can create an interference pattern that can be detected, and the actual volume of displacement can be detected as well. And so what this looks like with ERISM is you get this like a lighter color here that's showing a pulling force as well as a pushing force that can be detected. And our workflow for this is that we differentiated MPCs over 8 days, seeded them on the ERISM chip, transduced them with the fusion protein, and then looked at their actual pre-clustering and post-clustering. Our first was to look at actomyosin contractility. And what we did find is that myosin light chain 2 was actually activated as expected. With the CD16 clustering, we see phosphorylation of nephrin and phosphorylation of MLC2, showing that there is that actomyosin contractility, and this is quantified here. We then also looked at, we found that there was an increase in the filamentous over globular actin ratio. So this is using actin sedimentation assay, and with CD16 clustering, we found an increase in the pelleted actin, showing increase in filamentous actin. And this is the actual clustering and phosphorylation of nephrin for this experiment, and this is quantified over here. To then determine how this is spatiotemporally controlled, we use the ERISM technique. And using a rhythm, after CD16 clustering, what we see, and this is with the transduced CD16 nephrin fusion protein, there is a change in the podocyte structure. And using the arisen time lapse, what we find is this dramatic increase in pulling and pushing forces over time that slowly dissipates as the clustering is engaged. And then also we then did this with the Y3F nephrin construct as well. And dramatically that CD16 clustering pattern and the change in the displacement and forces was not observed. A time lapse is shown right over here. This is a representation of these. And as we see with CD16, it goes up. And this is not shown with Y3F. And this is quantified right over here. Now, in a previous paper, what they found was that these pulling forces actually co-localize with focal adhesions. And we also know that nephrin might be able to signal to focal adhesions through certain adapters like NTK. So we want to actually look at the focal adhesions as well to see if some of these force transmission occurred through focal adhesions. And so just looking at some of the markers of focal adhesion activation, first off, with nephrin clustering, we do see phosphorylation of nephrin, and this does not occur with the Y3F mutant. We then looked at paxillin at focal adhesions, found that it was increased with clustering, but not with the Y3F mutant. And then same thing with FAC, same pattern as well. And this is focal adhesion kinase and P130Cas. This is quantified right over here. So this basically tells us that focal adhesions can also be activated. However, we don't really know exactly whether it is downstream or upstream of that contractility and if it's dependent on it. But it's still something that's really important to note for podocyte biology. We then wanted to determine if this was relevant for an actual mouse model. So we used a Y3F mouse model and then looked at diabetic nephropathy, which has mechanical strain, to see if the disease is worsened. So we used what's known as streptozotocin or STZ to induce like a type 1 diabetic nephropathy or diabetes, which leads to nephropathy, collected urine on week 23, and then collected tissues for the assessment of the hallmarks of diabetic nephropathy. And what we find, our main phenotype, is that nephrin Y3F mice do have exacerbated proteinuria with diabetic nephropathy. This is shown right over here with an albumin gel and quantified with the albumin excretion rate. What we see is this dramatic increase in proteinuria, specifically in diabetic Y3F mice. However, looking at markers of glomerular filtration rate, within the diabetics, there was no difference in GFR. And these markers are inversely proportional to the GFR, meaning that there was a decrease in GFR over time. And so what we can say is that for the same amount of mechanical strain, there was a higher amount of proteinuria in these mice. And then we looked at certain markers of glomerular sclerosis and hypertrophy using periodic acid shift staining, which stains essentially matrix proteins and carbohydrates. And what we can see here is there is an actual increase in diabetic nephropathy and diabetic Y3F mice for both of these metrics, meaning that sclerosis and hypertrophy were both exacerbated in this disease. To look at things at the ultrastructural level, we also looked at TEMs, or transmission electron microscopy, and in the diabetic Y3F mice, they did have higher foot process effacement and GBM thickening. However, what was very interesting is that the Y3F mice on their own did have effacement and GBM thickening, and this was not necessarily made worse as they were diabetic. And so there seems to be some type of ceiling effect in which they can't actually efface any further. So what we're getting proteinuria, however, without that same kind of typical marker shown there. And finally, we want to look at to see if there is any potential for podocyte detachment in the disease. So we looked at podocyte markers such as WT1 shown here, and WT1 is a nuclear marker for podocytes, and then nephrin as well, and they co-stain, and you can actually detect the amount of podocytes and the density of podocytes with this pink staining. And what we see is there is a decrease in podocyte density in the glomeruli for diabetic Y3F mice. And to determine if this is actually related to the loss of podocytes, rather than just the expansion of, say, the glomerulus, we actually do see detections of podocyte proteins in the urine, so podocyturia, in diabetic Y3F mice. And this is quantified right over here. And so it seems that that contractility and mechanical signaling to the focal adhesions is important for the podocytes to then resist the mechanical strain and prevent detachment. To summarize once again, Nephrin signaling contributes to these adaptive mechanical responses in podocyte injuries, for example, diabetic nephropathy. When clustered at the slit diaphragm, nephrin coordinates actomyosin contractility and focal adhesion signal activation through these specific motifs, the YDXV motifs. Loss of the signaling of the tyrosines in vivo led to an exacerbation of diabetic nephropathy, for example, proteinuria, hypertrophy, and podocyte detachment. However, we did not see foot process effacement and GBM thickening, meaning that these specific tyrosines might be also important for signaling to these mechanisms as well. And for just a bit of a mechanism shown here, when there's tension or shear stress, you might get clustering of nephrin here. That engages actomyosin contractility, which then might be able to signal to the focal adhesions or back and forth here. There would then be an increase in the GBM, potentially. This is kind of more tentative. And then there also might be a signaling to actually create podocyte migration, which leads to that effacement. And when there's the Y3F, you don't get any of these changes, and you can get podocyte loss. And I would like to acknowledge the co-first author of this, Clara Martin. She has done a ton of work on this project. It is not just me as well. I really can't take all of the credit. Other co-authors are star asterisk right over here, and this is my lovely lab with the Nina Jones lab. Collaborators in Scotland, my supervisor, Nina Jones, scholarship funding through NSERC, grant funding, and my advisory committee. And I'll take some questions. Thank you.
Thank you so much, Casey. Such a wonderful talk and lots of really cool data. So now we're going to open up the floor. If anyone has any questions, you're more than welcome to raise your hand or drop them in the chat. All right, go ahead, Matt.
Yeah, really nice talk. That's really cool work. I have a more, I guess, molecular kind of question, and I don't know if I missed this, but is it known what the phosphorylation does? Because it seems, I don't know, it seems strange to me that these things would come together and then be phosphorylated, which if they're all phosphorylated, that would kind of be repulsive, no?
Mm-hmm. It's actually a very intricate mechanism. So in podocytes, that basolateral domain has so many other things going on. What we actually think is happening in clustering itself, well, this is kind of a bit of a debate because generally it's assumed that nephrin might be able to interact with the slit diaphragm regardless and creating this clustered pattern at baseline, and then that the phosphorylation of nephrin is continuous, it's always going on. But there are other models that actually say that nephrin only clusters upon certain stimuli. Personally, from this data, it seems that there's some kind of stimulus that then creates that phosphorylation. But what I think might be happening is that there's a bunch of kinases that are present in that domain. And there are these lipid rafts at the basolateral domain, and it's regulated by another protein known as podocin. Podocin then maintains these lipid rafts. And so when all of this comes together, it seems that all of their affinities and it just essentially everything is in proximity. And so it leads to that phosphorylation itself. Some of this is also stabilized by phase transitions as well. And I think that there's a little bit of more competition of what can actually be present and what is more likely to bind in a specific context. Because there's a bunch of other interactors that might be able to interact, and when it's clustered, maybe the more the interactors that have a higher affinity might be able to come in those instances. But yeah, it's a very complicated mechanism and it's hard to explain in such a short time.
Yeah, for sure. That's cool. Thank you.
Awesome. So I'm not seeing any other questions, but you're always welcome to reach out to Casey afterwards.